All right. So, hi. Okay, I'm recording this time. Good, because I just did this whole presentation and it wasn't being recorded. So, here we go. Round two. Um, we are going to talk a little bit about protein synthesis today. You guys should have a note packet if you went to school that says gene to protein. I would have that out beside you, and you can kind of go follow along with the slides, fill in information, add additional information, kind of as usual. If you, for some reason you can't get to school and get those notes, I can send you a copy. Just let me know, and then you can print it off at home. All right. What else did I want to say before we got started? I am going to probably talk pretty quick, but I feel like you can pause, you can repeat, you can take some breaks. You don't have to just sit here and listen to me go all the way through. I am going to try and go pretty quickly, so it's like hopefully a 15-minute video, and just over transcription today, and then the same tomorrow for translation. Okay. If you have any questions, concerns, want me to repeat anything, just post it in the comments below, and I will try and address those tomorrow in tomorrow's video. All right, so here we go, gene to protein. First off, why are we talking about protein synthesis in a genetics unit? And I think you guys all know that. It's because it doesn't matter what your genes say if you don't make it into a protein, right? Proteins are the actual expression of your DNA that will then cause the phenotypic result in your body. So, for example, someone's hair that has gone gray still has the gene for brown hair. They're just no longer expressing it. They're no longer making the proteins that will actually make their hair brown. Their DNA didn't change. It's just the expression of that DNA into proteins. Okay. So that process is sometimes referred to as the central dogma because it is so important to biology. The DNA getting transcribed into RNA, which is translated into proteins that makes our phenotypic result, our trait. Okay, and that should probably sound familiar from biology. And we're just going to get into more depths of it in AP. All right, so metabolism was probably what first started us thinking about the result of something going wrong in our DNA as far as like an effect on an enzyme or a protein. Okay, we saw that when there was changes that something didn't function right somewhere in that metabolic pathway, and you no longer had the appropriate result that you needed to function. Okay, so for example, you lack a specific enzyme where you are now intolerant to something. Like the classic example I always bring up is lactose intolerance. Okay, you don't have that enzyme for that metabolic pathway. Now you can't break down lactose. We've talked about a few others like albinism. You lack the enzyme to make melanin, and therefore you don't have melanin in your skin. You don't have pigmentation. PKU was that amino acid that the mutation causes you to unable to break it down, so it would build up and could actually kill you. But if you're tested right away, which you are if you're born in a hospital, then you would just have a diet low in that amino acid and you would be fine. Tay-Sachs, unfortunately, is the inability to process lipids, and they build up around the brain. And if someone is diagnosed with Tay-Sachs, which would be inheriting the recessive allele from each parent, they won't probably live past 5. It's very sad. Okay, this idea of one gene controlling one enzyme was first established through a lab that tested a particular mold. It would grow on minimal media, okay, meaning it just had a few nutrients. But then they exposed the spores to x-rays and caused all sorts of different mutations. And different mutations would cause those particular spores, maybe they could still grow on a complete medium, but if you put them on the minimal medium, they couldn't grow anymore. And so then they would add just specific amino acids or nutrients and figure out what that one enzyme they were missing was, because they couldn't make that one particular nutrient. And then that changed to the one gene, one protein, because not all genes do enzymes, right? They code for proteins in general. And that's gave us a pretty good idea of how genes code for proteins, which cause a result. We've now, of course, found exceptions that make it a little bit more complex, especially for like proteins that have more than one polypeptide that make them up. But it's a good starting point. Okay, so today we're going to talk about the first step in protein synthesis, which is going from DNA to mRNA in the nucleus, and we call that transcription. Okay, we'll look at the second half tomorrow. All right, so transcription, DNA to RNA, specifically mRNA. And you guys know the differences between DNA and RNA by now. We have the ribose sugar versus deoxyribose. We have uracil instead of thymine. Okay, and it's single-stranded, which makes it possible so it can leave the nucleus, which DNA cannot leave through those nuclear pores. We keep it protected in the nucleus. It makes it be able to actually behave in many different ways. So RNA is very functional in our cell. The mRNA, taking that message from the DNA to the ribosomes, but a couple others we'll talk about are tRNA in translation, and then rRNA, which makes up the ribosomes. Another one we won't talk about too much is siRNA, that kind of like can attach to viral RNA and kind of help in disease prevention. All right, so for transcription, we need to take that DNA code for the gene and change it to mRNA so that code can go out to the ribosome to make our protein. Okay, for that to occur, we need to copy what we refer to as the template strand. Okay, that is the strand that is actually going to be matching up with mRNA to make the complementary sequence. Okay, the other side we call the coding strand, which is not copied into mRNA, and it would have the same sequence as the RNA, except for the have T instead of U's, where the template is complementary. RNA is built just like DNA, where it only goes 5' to 3'. You only add on to the 3' end. And the enzyme that makes that possible is RNA polymerase, represented by that big pink bubble. All right, so we have a bunch of RNA polymerases, just like we have a bunch of DNA polymerases. The one that we're concerned with is RNA polymerase 2, because that is what transcribes DNA into mRNA, which is what happens in transcription. Okay, how it does that is it looks for the promoter region. That's the chunk of DNA before the gene of interest, before the actual sequence. Okay, and that's where the RNA polymerase 2 is going to bind. Okay, it looks in that promoter region for a specific region called the TATA box. Now, I've heard many people call it TATA box, but I just can't do it, so I call it TATA box. And it is actually a sequence of TA, TA, and then usually followed by some A's. Okay, this is where the RNA polymerase will attach and then move towards the DNA that actually needs to be coded. Sometimes there's also in eukaryotes a promoter region, an enhancer region far, far up from the promoter region that can actually turn up transcription, make this happen more frequently, more quickly. And that can be very far upstream, hundreds, thousands of base pairs, sometimes even on a different chromosome. Crazy. Okay, so where the RNA polymerase 2 attaches, it has a bunch of other proteins, sometimes even hormones, other things that help it attach and function correctly. We lump those all together as transcription factors. We're not going to talk about specific ones in here because there can sometimes be up to like 35 other proteins working to help RNA polymerase 2 attach and function correctly. So just understand those are present, but we're not going to get into them too much. But they help trigger this whole process. Okay, so now RNA polymerase is cruising. It's adding mRNA complementary sequence 5' to 3', adding onto the 3' end. Okay, U is attaching to A instead of T, right? Okay, now for prokaryotes, that can kind of be it. Now, eukaryotes, we are a little bit more complex. Okay, first off, that chunk of the template strand that we just copied actually isn't exactly what we want to read when we get to the ribosome. There are portions of the gene spread throughout that DNA, and there's portions of junk, we call it junk, just extra DNA, within the portion that will code for the appropriate protein. So the bits of DNA that actually need to be read by the ribosome are called exons. They are going to be expressed. And the bits that we don't want are introns. They are in between the coding sequence. So we need to cut and get rid of those. The DNA that we have just copied into mRNA, okay, has transcribed both the exons and the introns. Okay, we call this mRNA the pre-mRNA because we need to get rid of the introns, and we need to do a few other things as well. Okay, so to get rid of those introns and make the mature sequence, as you can see on the bottom, which is typically much, much, much shorter, we've got a lot of introns to cut out. We will need to what we call splice, okay, this pre-mRNA. Okay, to do that, or actually, I'll show you how we do that in just a second, but first, just to understand how important this is, okay, if you don't splice exactly correctly, okay, you can throw off the entire reading frame. Meaning, if you cut one too many bases out, everything is going to shift to the left. And so those 3 mRNA codons that you used to code for a specific amino acid, which we'll talk about more in translation, okay, everyone after that point is going to be off. It's going to be different. Okay, so we're going to basically mess up our whole protein. It's probably going to be non-functional. Okay, so you have to splice exactly correct. Okay, and so we use what are called SNRPs. I love that, I think, because it reminds me of Smurfs. Okay, these small little RNA chunks that fold up just so with some proteins as well. And they kind of act like an enzyme, which is sort of weird. But RNA, because it's single-stranded, can actually fold back and bond with complementary bases in different sections. So it can fold in a specific shape, just like an enzyme. So it can function like an enzyme. So these little SNRPs combined with other RNA with protein, and then a handful of them all together make up a spliceosome. And these are like our little scissors that will go along, and actually I should probably say scalpel, because they cut at very specific spots to get those introns out without taking any of the extra DNA, the exons that we want to leave, okay, so that we don't mess up that reading frame. It's kind of crazy. All right, so we do this whole cut and paste thing with our spliceosomes so that we end up with the specific exons, and that's it, that can be read for a gene. However, to make things even crazier, the same chunk of DNA can be spliced differently to make different proteins. Okay, same chunk of DNA spliced differently will give different exons, right? To be expressed, to be read. And so it makes our DNA almost infinitely complex, because besides being 3 billion base pairs long, we can also cut it differently. And so, yeah, it's just wild. It's like mind blowing. What else has to happen to mRNA before it can leave the nucleus? Well, we have just our exons now, but we need to protect it a little bit. We put a 5' GTP cap on one end. Oops, sorry. And that's the end that's going to first enter the ribosome. And then we have a 3' poly A tail, and that tail can actually change in length. The 3' end is the end that's going to be attacked by enzymes to start degrading that mRNA. So if we put a long poly A tail, we can actually use that mRNA sequence multiple times in the ribosome to make up multiple proteins before it gets fully degraded and broken down. And so maybe a protein like hemoglobin that we're making all the time, well, let's make a long poly A tail so we can use that mRNA multiple times before we have to go back through the whole process of transcription. All right, so we just talked about transcription, going from DNA to mRNA. That all occurs in the nucleus. Now we have copied the code for our gene, and we can take that out to complete the process of protein synthesis, which we will talk about tomorrow, which is the translation of that mRNA into an amino acid sequence. All right, so if you have any particular questions or want some clarification on any of that, please, again, put that in the comments below. Otherwise, oops, what? No. What's going on here? Okay, whew. I thought it cut me off. Okay, anyway, have an awesome day. Talk to you soon.